to you today about a technique we've been working on to take time series expression experiments and to extract from them a set of predictions about transcription factors that are working in combination in order to have regulatory effects in these experiments. So to begin, I'm going to talk about the kind of dynamic models you can build from time series expression data, and then I'll get to how we actually need to change these models in order to take into account the combinatorial structure of the regulation which is occurring. So what we do when we want to create these models is take some kind of time series of expression experiments, either RNA-seq or microRNAs, and when you have just that data, it looks like this in the bottom. And it looks like a mess, of course. So the x-axis here is time, the y-axis is differential expression, and when you plot every single gene, just by default, there's not a lot of information there. And the goal is to actually come up with triggers to figure out why groups of genes are acting together, and the way this can be done is by adding additional information. And so the additional information I'm going to use today is various kinds of static information about how transcription factors and genes interact. So the major kind of most recent source of these are, for example, ChIP-seq experiments where you can see transcription factors binding up region of various genes. So once we have time series expression data as well as the static information, we can begin to build models. And so imagine we have this toy experiment here, and you can see that the genes have been color-coded based on their regulators. And the experiment here, of course, is very, very simple, but there are regulatory events that occur over time. And the idea is that transcription factors are driving these. And so we can build a model that looks like you see in the bottom left. And in this model, we can actually identify split points where some amount of transcription factor activity has caused some genes to change their expression, in this case up, and other genes to change their expression down. Once you've actually built this model, then you can do some statistical tests and actually figure out, well, which of these transcription factors were actually active at each of these time points. And so at the moment, we're just modeling each transcription factor on its own. And to kind of give you a better feel for how this works, imagine we're looking at a single gene here, let's call it gene X, which is in this green group. We model the activity of gene X through this model using a hidden Markov model framework. And so at each time point, the gene will emit some value equal to whatever we measured it at in the time series. And when the gene transitions, it does so based on a set of split points that we learn. And on each of these split points, we look at the transcription factors that bind the gene. And so gene X, we know, is bound by transcription factor B. And so if we see a bunch of genes bound by transcription factor X that are going up, when we try to model gene X using our model, we will see that we expect gene X as well to be expressed. So that was an example on very toy data. So this is what this looks like on a real experiment. So this is yeast data, and again, you can see that things are much more complicated, but you still see patterns where we can find these split points. We can identify transcription factors, which are statistically overrepresented in the genes going either up or down. And at the end, you kind of get these groups of genes that when you reach some steady state after an experiment, you can analyze for some kind of similarity of function, given that we expect they've been regulated in a similar way. So at the moment, we're using a very simple additive model of transcription factor regulation. And so if you see something like this, where you have two transcription factors A and B, maybe transcription factor A is active and transcription factor B is not. And when you have a gene which is annotated with both transcription factors A and B, well, it'll be expressed, and that's great. And what you see here is essentially a logical OR relationship, right? So one of the transcription factors is active, the other is not, but that's still sufficient to make the gene expressed. But this is a simplification, of course, of what's actually going on. Many times you actually need to have multiple transcription factors acting in concert. And so you'll see something like this, where you have transcription factors A and B, now both of them active. And even though they're active, there's some number of genes which are bound by transcription factor A and some number of genes bound by transcription factor B that are not active. But when you look at genes bound by both A and B, well, they will be active. And perhaps what's going on here on a biological level is A and B are forming some kind of transcription complex, which drastically upregulates the expression of genes bound by both of the transcription factors. And so this kind of model, this complexation of relationship, can be thought of as an AND relationship, where in order to have a gene expressed, you need to have both of its constituent transcription factors active. So before I go on to exactly how our method works, I wanted to mention a few things. First, I'm going to talk almost exclusively about pairs of combinatorial transcription factors. And that's simply for the matter of time. Our method can also be used on combinations of more transcription factors, triplets, or even larger groups of combinatorial regulators, if you believe that that's actually what's going on in the biology. The other thing that our model does, it can explicitly model AND and OR relationships. And again, today I'm only going to talk about AND relationships. However, additional logical functions could be modeled as well. If you think you have two transcription factors that work in an XOR relationship for whatever reason, great, you can model them. The last thing I want to point out is that since, again, we're using dynamic models, we can actually predict for each of our combinations the condition and the time point at which the combinatorial binding occurs. And this is great if you, when you want to eventually validate things. It provides you a much more selective kind of experiment to run. So in order to actually build regulatory models which take into account combinatorial interactions, we use a three-step process. The first thing we do is take the huge space of possible combinatorial pairs or triplets or whatever, and prune them into a smaller number of possible pairs or triplets that we can actually interrogate using a model. Once we've pruned our space of possible pairs, we actually build a regulatory model which is aware of the relationship between different transcription factors. And then once we've built these models, we have to extract from it some number of predictions. So the first thing we do is reduce the number of possible pairs that we look at. And if you look at the numbers on this slide, I think the reason we have to do this is quickly self-evident. When you look at yeast with 200 transcription factors, perhaps we could actually interrogate all 20,000 possible pairs in a principled framework. But when you go to human with many more transcription factors, looking at 2 million pairs of transcription factors is very difficult to actually interrogate in a principled way. Similarly, these are just pairs. If you go to triplets or larger combinations, the space of possible combinations is enormous. And so what we have to do is come up with a way to just find some number of possible pairs that we think might possibly be interesting. And so the way we do this, imagine you have two transcription factors, again, A and B. We perform a simple statistical test where we look for the over-enrichment of genes in the overlap of these two transcription factors. So again, we have a list of all the genes regulated by A. We have a list of all the genes regulated by B. And if the number of genes which they regulate in common seems to be statistically significantly large, then we say, OK, this is a potentially interesting pair. Let's add it to our model. And just to give you a feel for when we're actually building these models, how many pairs we take into account, we've run this using on the order of 600 to 1,000 different pairs being interrogated at a time. So even though it's much less than this, this actual number, we can still look at many different things at once. So once we have actually identified some number of pairs that we think might be interesting, we want to develop a modeling framework so that we can actually figure out which pairs might be active. And we do this using a technique we call cDREM, which is a combinatorially aware version of the dynamic models which I showed you previously. So I mentioned that we have these two transcription factors, A and B. And we found that this combination of transcription factors is interesting because they have a large overlap in the number of genes that they regulate. So one thing we can do is, in order to express this, create virtual regulators. So let's create this new transcription factor called AB. And this virtual transcription factor regulates all of the genes shared by A and B. So we can do this for all possible pairs of transcription factors we think are interesting. So we looked at A, B because they have a big overlap, but additionally there are many other such pairs which are interesting, AD, BD, etc. And the thing that starts to happen is that the structure of these virtual transcription factors falls into a kind of grouped framework that it gives us a structure to the problem. And so how we're organizing this is, so we have this group A. And so what is group A? Well, it's all of the virtual regulators that contain transcription factor A. So of course, transcription factor A itself, as well as AB and AD, et cetera. And we can do this for every transcription factor. So there's a group B containing all the regulators that contain transcription factor B, et cetera. So now that we have this group structure, we want to actually be able to represent this in our models. And so the way we do this, I mentioned previously that when we have a split in the model, some amount of genes go up, some amount of genes go down, and the way we actually predict the transition probabilities for genes is based on the transcription factors that regulate them. And we do that using a logistic regression model. And so this is what this looks like. So there's just a typical logistic regression objective function here, and a Lasso penalty term on the weights. And these weights are the weights of a transcription factor causing a gene to go up or down at a split. So what we want to do now is find a way to encompass the group structure of this new set of regulators that we've built. And the way we do this is using a framework called the sparse group Lasso. And so this is what I'm showing here. And what you can see is it very much resembles the typical Lasso, but it has an additional set of penalty terms. And these penalty terms are a set of terms over the groups. And so I showed you previously group A and group B, and those are the groups that we're actually penalizing over. And so what is the structure of this penalty like? So just to give you a kind of intuition about it, you can take a look at this plot here. So imagine we have two weights, and these weights are representing transcription factors in the same group. If you have a typical L2 penalty, well, you just get a circle of equal penalty contour. If you have an L1 penalty, you would have the diamond shape. With a sparse group Lasso framework, you actually get something between those. And so what you're seeing is some amount of sparsity, the less that you would see in Lasso. Now, this is still within a single group. When you look between groups, which you can't really see on this very simple two-dimensional plot here, you actually get the sparsity that you do see in Lasso. So this penalization structure gives you sparseness between groups, as well as some sparseness within groups. And again, this is useful because when we have these groups of transcription factors, we would expect that, again, we want substantial sparseness between groups because it's a very strong statement to say both A and both B are functional. But it's a less strong statement to say once A is found to be functional that perhaps AB is also functional. So once we've built models using this new penalization structure, we have to extract predictions from it. And the way we do this is by looking at the weights assigned by the logistic regressor and combining this with the hypergeometric p-value that you get at splits in order to see if, again, things get regulated up or down. So here's an example of what this looks like. I'm only showing you here the predictions that we make that are AND regulators. Now, when we build these models, we use individual transcription factors as well as ANDs. And also, we're explicitly modeling OR relationships. But I'm just showing you the ANDs here. And all of the blue stars here are things that have been annotated in the literature and known to be true. And so both of these things that I'm showing you here are perturbation experiments in yeast for some kind of stress. And you see kind of known heat shock combinations come up, such as FKH2 and MCM1, which is related to transcriptional activation. You see that this gets repressed early on in heat shock, which makes a lot of sense based on the biology. So that was a qualitative look at what these things are. But we can also use various quantitative measures to figure out if we're actually doing something intelligent when we're actually finding our sets of pairs. And so one way you can look at this is saying, for the various pairs that we have annotated, do they exist with other annotations that exist in the literature? And so specifically, we look at BioGrid, which contains sets of protein-protein interaction. And if you take 2 transcription factors at random, you find that there's about a 5% chance that their activity has been annotated as existing as a pair in BioGrid. If you just perform our pre-processing step, which is just, again, finding all transcription factor pairs, which are past some statistical threshold of having similar genes regulated, about 18% of those are found in BioGrid. If you take the output pairs using our modeling framework, it turns out about 30% of the pairs in our model are actually found as annotated. So at each step, you get an enrichment, which is what you would expect to see if we're doing something correct. There are other things that we can do to quantify these pairs, but in the interest of time, I will skip over this. Feel free to ask if you're interested. And then the last thing I want to point out is that not only I've looked at yeast experiments, shown you yeast experiments previously, but we can also run this in human data. So here's an example of a human immune response experiment. This is an individual who was exposed and ended up getting the flu. And again, I'm just showing you the AND pairs that show up. And a lot of these are known. For example, things like STAT1 and IRF1, which are well-known complexes which show up at the beginning of an immune response. But there's some other interesting things that show up. So our relationships are logical. We're looking at the logical relationship between 2 transcription factors. And so that's not necessarily a complexation. So one of the things that shows up in our model is IRF1 and IRF2, which is interesting because they're actually known to be competitive binders. So IRF2 will repress a lot of the IRF1 targets. And so what we're actually probably seeing in the model with this pair is that genes which are regulated by both IRF1 and IRF2 suddenly become repressed as soon as IRF2 is active. And this would occur in contrast to whatever activity is found in genes bound by IRF1 but not IRF2. So an interesting relationship, not necessarily a complexation, but still probably true. So that's all I have for you today. So what I've done is shown you a method which can take the huge space of possible combinational groups and whittle it down to a small number of condition-specific predictions of group combinatorial regulation. We've taken a look at validating this both quantitatively and qualitatively in both yeast and human data. And that's it. Thank you very much.
Thanks, Aaron. I think we have time for one question.
Hi, Lana Kamer from University of Hawaii. Thank you for the talk. Recently, we are actually exploring the tool, your sibling tool called cDREM. Where I found, could you go back to the second slide that you showed on the the tree structure?
Okay.
The second one. Yes. So this kind of decision points, how are they made and do they correlate with the expression profile change? Does that indicate if, let's say if a transcription factor pops up or microRNA pops up, does that have any relationship with if there is a transition point for this transcription factor itself, expression change or microRNA expression level change? So I found it was hard for me to understand that this kind of decision making for the splitting. That's one thing. Another thing is you have some on the y-axis the tree goes both on the positive and the negative directions. So what does that mean?
Mm-hmm. Sure. So to answer the first question, does expression result change whether we predict something to be active or not? And the answer to that is for transcription factors, we do not use the expression of that transcription factor as an indicator of whether that transcription factor is active. In the past, we've looked into that, and it turns out that they don't tend to be a very good predictor. For microRNAs, when you're looking at cDREM specifically, in fact, I believe we do use the microRNA expression level. It does seem to be more predictive. So the second question, what are these, how do you get negative values? So all these axes, they're expression, but they're differential expression. So everything is normalized at 0 at the beginning. And so as things move through the model, whatever basal rate they had at the beginning, you can actually go down from there. And if something is repressed, that's actually represented as a negative value on the axis.
So the branches are below 0 are the events, the transcription factors or microRNAs that are repressed.
Right.
in that time point. Okay. Also, you're talking about this combination for the transcription factors. I can imagine there are many other mechanisms that can potentially regulate the gene expression, but besides transcription factor, besides microRNA, there's also DNA methylation, histone modification. So are you guys gonna pursue those directions as well?
So that's really, as long as what we say something's a transcription factor, part of this work was creating the idea of a virtual regulator, right? Where you can create essentially transcription factors that represent either the combination of different transcription factors or whatever. You could imagine that these virtual regulators could actually be anything that you believe to have regulatory activity. And so could you use a methylation signal or something like that? In theory, you could. That's not something we've pursued simply because the data is
So I can
less
just
abundant.
change your input matrix, right? And then still use that.
I think we have to proceed, and you can take that offline.
Can we talk afterwards?
Yeah, certainly.
Thank you, Aaron.
Thank you.
Thank you.